I thought this would be interesting to talk about the role of diet, specifically in IBS, because that's where most of the studies, but don't think that this is only related to IBS. But that's where most of the studies have been done. And I purposely ended the last talk with the role of Mediterranean diet and low FODMAP, because there have been a lot of studies that have happened recently, and I'm going to try to bring you all up to date with regard to the recently published literature on diets in IBS and specifically. But FODMAP and Mediterranean, at the end of the day, those seem to be the ones that most of the data has been focused on. So we're going to go through this in a little bit more detail. That hasn't changed. And we're going to start by talking about the role of foods in general and the association it has with the developing IBS and the role of it causing symptoms. These are actually two different things. So the development of IBS and the generation of symptoms. And I'm going to talk a little bit about the mechanism of what we know about the role of the pathophysiology of how foods may cause symptoms. Again, that's not specifically related to IBS, but just how it may cause symptoms in general. And then we'll end by going through a number of different studies. And what you're going to see is that I'm really going to focus on things that were just published. And most of this is brand new because this is a really new area for all of us. So most of them are published within the last 6 to 12 months that I'll be showing you today. And I'm not even showing all of the data that's been there. So this is a busy slide, so don't spend a lot of time looking at it, but there are actually two different studies here. And they're relatively similar. That's why I put them together, and I just want to highlight them. They've received a lot of lay press, both of them have, and so it's worth just reviewing them. And I put them together because they use the same cohort of patients or individuals. They used the UK Biobank to look at it, and they asked very similar questions. So on the left-hand side, they said they looked specifically at the role of processed foods. And there's a whole definition of what is processed and unprocessed food. Basically, if it comes in a box, it's processed. And if it doesn't come in a box and you buy it off a shelf and you make it yourself, it's probably unprocessed. It's more complicated than that, but I think you all get the thing. And you can see here some pictures. This is what they called ultra-processed foods. Again, you can see that it has sodas and fast food, et cetera. And again, a whole definition, but I think we all sort of understand what it is. And so basically, they looked at a large number of people that were in the UK Biobank. In this case, they had, I don't know if you can see it, but over 178,000 people in the biobank. And you have to know, one thing you have to know about the biobank is that they were an older group of people. So the mean age was 55 years. In both groups, they're about the same age because they use the same cohort of people when they started the study, when they first entered into this study. So these people were entered in around 2009 to 2012, and then they ended up following them for 11 years. So what they looked at was, and they had an extensive dietary history. On the left-hand side, they looked to see, well, what was the people that were eating unprocessed food or ultra-unprocessed food, what were the odds that they would develop IBS symptoms? So that was the question they asked. So they had no history of GI issues, no diagnosis of IBS or other GI issues. And what they found was that about 1.7% of people, so low number, developed IBS during the 11-year follow-up. And that was significantly higher than people that did not eat ultra-processed foods. And so that's what basically came out of that. And you can see that about 21% of their food intake was related to ultra-processed food in this population. So the bottom line was ultra-processed food could cause IBS at a much higher rate than eating unprocessed food. On the right-hand side, this one actually received much more attention in the lay press because it looked at 5 healthy behaviors, which was kind of interesting. Never smoking, optimal sleep, higher level of rigorous activity, moderate alcohol intake, and higher dietary quality. So obviously we're talking about the dietary qualities. And they did basically the same thing. They looked at these patients, followed them up for 12 years, looked at who developed IBS, and you can see that for foods, it's right here, it crosses 1, so it's not that significant. Really, optimal sleep was the one that came out. But what was more important in the study was that they looked at the cumulative number of healthy lifestyle behaviors. And what was most interesting was the fact that so few people had 3 to 5 lifestyle behaviors that they had to lump them together, because most people had 1 or 2 at tops lifestyle behaviors. And you can see that if you had 3 or 5 healthy lifestyle behaviors, that was a protective for developing IBS. So the bottom line was, yes, you can kind of prevent IBS or alter GI symptoms if you live a relatively healthy lifestyle. And again, that's what, in doing all of these things, none of them individually except for sleep, but combination may be important for it. So again, both of these received a lot of interest. Food is clearly related. Ultra-processed foods may be important as well. So we do know that foods can have a tremendous effect on the GI tract, and I'm just going to highlight 2 studies that have shown that. The top one uses confocal light endoscopy, and it's not a widely used technique, but it shows some interesting things. So this is a healthy duodenum, and you can see an increase of lymphocytes in this one, and then the white here is increasing permeability. You have to take my word for it that they saw an increase in permeability, an increase in intraepithelial lymphocytes associated with it. What they did in this study is they put food on the duodenum. So they did baseline with confocal, then put food, specifically wheat, yeast, milk, soy, and eggs. You can only do a limited number of foods, and you can see that of them, 61% of people, and these were IBS patients, had one of these abnormalities, increased permeability or lymphocytes or both, versus yeast, milk. You can go down the list and show that. And that's within a few minutes after placing the food, suggesting that in those patients with IBS, that perhaps those type of foods are the trigger of symptoms. The second study is really a landmark study, which I'm going to show more details later. And this was just a small part of it. I'll show you the bigger part of the study later, but they looked at a small number of IBS patients. And what they did is they injected a couple foods into the rectal area, submucosally, okay? And so they have saline as a negative control and histamine as a positive control, healthy volunteers in IBS. And you can see negative, negative, positive, positive. The white markers represent the edema that's occurring. So you can't see it that well, but you can see that for the healthy controls, when they injected soy, wheat, gluten, or milk, there's really not much going on. But for people with IBS, that almost all of the IBS patients had reacted to at least one food, where only 2 out of the 12 healthy volunteers. Again, suggesting that these foods may be a trigger of symptoms in some patients, and I'll show you some of the pathophysiology in a minute. And just to confirm this, there's been a meta-analysis that looked at the role of increased permeability among IBS. And the left-hand side just shows you a figure. This is the percent that have, in the studies, that have had increased permeability, and this is diarrhea, IBS with diarrhea being the most predominant, but you'll notice that even in constipation, post-infectious IBS, mixed IBS, that there's a fair number of patients, not all, but a fair number of patients with increased permeability. Increased permeability was associated with bowel disturbance, pain, overall severity, also depression and quality of life in some of these studies. So again, suggesting that this may be an abnormality that may be relevant in some, but not all patients. And as I showed you earlier, some of this is related to food that we may be taking. And this sort of nails it home with regard to the role of permeability in IBS. And this is a study that was done by Nick Vern's group, where they gave L-glutamine. Now, the important part about L-glutamine, and you know this from the ICU, that you use that to reduce permeability in the GI tract, where patients that aren't eating have increased permeability, they're more susceptible to infection, so we give them L-glutamine. In the same case, in this study, what they did, they took IBS with diarrhea that were post-infectious. Half of them had increased permeability. They enrolled them into this trial of glutamine versus placebo, and what they found was that there was a dramatic improvement. So 79% of patients improved in the glutamine group and only 5.8 in the placebo group. That differential is enormous. The editorial said maybe too good to be true, but it needs to be repeated. But what was most important to show here is that a strong correlation existed between improvement in permeability and improvement in IBS. That was the sort of the take home. Hard to see, but the correlation was strong in this case. So suggesting that permeability may be involved in some patients with IBS. So this is the study I want to talk a little bit more about, the landmark study that I said, and this is not specific to IBS, but this study, which was published in Nature, showed a mechanism whereby food may trigger abdominal symptoms. Again, not specific for IBS. Most of the study was done in animals, but it shows an interesting pathophysiology. So in the steady state, we eat food, and there's reactions that occur in the GI tract, but we're all protected from it, and it doesn't cause increase in symptoms. But you can have a breakdown in what we call oral tolerance. And in this case, it was due to an infection. And they gave a bacterial infection to the animal, increased permeability, the antigen in the food, then they fed the animal a protein. That protein, there was increased permeability, and so there was a reaction that occurred, and most of the studies showed that overall reaction, proving it pretty definitively that this is the mechanism whereby the increased permeability could cause symptoms associated with the food. So basically what happened was that there was an increase in specific IgE levels that were not found in the serum, only localized. These triggered a reaction in the mast cells, and that mast cells activated the afferent neurons. Later on, after the animal recovered, they reintroduced that that protein, and they saw the same type of reaction occur. So that was the idea behind it. You can become sensitized to a food, which may explain why sometimes patients will say to you, this started, a year ago, whatever. Now I can't tolerate a variety of different types of foods. This may be the case behind it. Another mechanism that was shown, and this is going to lead into the low FODMAP, and this was done by Prashant Singh's group, published in JCI, which is shown here. This is the group from Michigan that showed it. And basically what they did was they took IBS patients on a high FODMAP diet or on a low FODMAP diet, took their stool, infused it in an animal, and then showed what happened with it. And basically what they're showing here is that there's an increased amount of histamine that's being released in patients with IBS-D, and then with patients when they're given a low FODMAP diet, it goes back down to baseline with it. And then on the far right, on the far one, they're showing that if they block the TLR4, toll-like receptor 4, that's the receptor that bacteria uses, that we use for innate immunity to prevent bacteria from infecting us, that you get, if you give an animal that's defective in it, you don't get the increase in histamine. So basically what they showed was that the mechanism that occurs from FODMAP is related to LPS via TLR4 in animal model. So interesting, because it's human stool that induced this from a high FODMAP and then a low FODMAP diet, suggesting that this may be the mechanism behind it. Again, bacterial induced. Both of these studies show those bacterial induced. So how do foods cause symptoms? I showed you two potential mechanisms, and the answer, I think, is that it's quite complicated. And as you can see here, food is complicated. We have a variety of different types of foods, and there are direct effects, and then there are secondary effects that occur from the foods that we ate, and even what we call tertiary effects from the psychological effects related around food. So it's not going to be just one mechanism. So we know if we are lactose intolerant, and of course, if we drink milk, and we'll get a fermentation byproducts that may cause symptoms. Again, remember, there are a lot of different effects that can occur to food. We just showed you two potential mechanisms, and here are a few others. So what about some of the data that exists with this? And in IBS specifically, this is just this is two different studies. This is from Mike Camilleri's group, probably the best run trial to date with gluten-free diet in IBS, 23 patients in each group with gluten-free, with IBS. They were given either a gluten-free diet or a diet containing gluten. And just looking at these figures, you can see that overall there was not a significant difference with it. But what he did show was that if you have the antibodies to celiac disease, you are more likely to develop symptoms, to develop improvement with it than if not the antibodies, if you had the gene, you're more likely to have improvement in symptoms than if you did not. There are a couple other studies, and basically not showing a significant difference. Gluten-free diet is generally not recommended for IBS based on this. There's one other study that looked at the low FODMAP diet first, followed by gluten-free. And what you see is that most of the improvement includes with the low FODMAP and not much beyond that. And of course, low FODMAP is low gluten, but does contain some gluten. And so this is more historical because we don't tend to recommend it for most of our patients. This is going to sort of review what we know to date up to the time this was published, and then we'll talk about the more recent studies. And this is a network meta-analysis looking at the different types of diets and the effect on IBS. And the bottom line, of course, is low FODMAP is the most effective of the diets. There's another diet called NICE, which is a European diet, very basic diet that's more advice on how to eat right. And you can see there that it crosses one, but does show some improvement. And other ones, like the sham dietary advice, alternative dietary, really haven't shown it. Of course, if you're given high FODMAP, symptoms tend to get worse, confirming the low FODMAP. So to date, the evidence sort of supports the low FODMAP diet, and that's where most of the studies have been done, and this is why you see it recommended the most for IBS. So this shows you the low FODMAP diet, the recommendations for it. I think I shut that off. I guess I did not. You could help me out with that. So that shows you the recommendation. What I want to point out, though, are the 3 steps of the low FODMAP diet. The first step is, of course, avoiding the FODMAPs and going to low FODMAP diet. But one thing we don't recommend, what we don't tend to recommend or is forgotten, is the phase 2, where you re-challenge patients. So usually it's 2 to 8 weeks of a low FODMAP diet, being very restrictive, and then re-challenging patients with the different types of foods. We're going to talk about which foods may be the better to challenge them with, and I'll talk about that in just a minute. And then long-term maintenance. All the studies are right here. They're all in the first phase. They're not on the second and third phase. We're really lacking data on what happens to patients long-term with the low FODMAP diet. But again, I want to emphasize the fact that you're supposed to re-challenge them and do that in an orderly manner, but we'll talk a little bit more about that in a few minutes. So this shows you the results of 2 different studies, and I put them again together because they're very similar. And these are really hard studies to do, but they re-challenge patients who have responded to a low FODMAP diet. Both of the studies ask the same question, which is, are there specific groups of the FODMAPs, right? There are 5 different groups of FODMAPs. Are there specific FODMAP groups that are more likely to trigger symptoms? Okay, so patients responded to FODMAP, and then they were randomized to receive each of the groups in different ways. And I won't belabor the point, but these basically try to show you the different manners. They all basically do it in a randomized way, give them a break in between, and then randomize them to a different FODMAP. And patients are blinded to it. The problem with this is that it's done differently. The monofODMAP reintroduced, and the specific type of food is different in the group, so it's hard to make definitive conclusions between the 2. But as you can see, this is sort of baseline, and then this is the reintroduction phase, and they're the 2 groups that seem to be have the most amount of response are the fructans and the galactans in the Michigan trial. In the European trials, it was the, as you can see, the fructans as well as mannitol seem to be the ones that higher. So both studies show fructans as being a major trigger for it, of the groups that be the major triggers associated with reintroduction. So this led to a study that was just published, it's in press, done by the Michigan group, looking at what's called the FODMAP simple diet. So it took their results and said, okay, if it's the galactans and the fructans, what if we made a diet of a low FODMAP, of just low fructan and low galactan, but having all the other FODMAPs present. And that's what this study did. And it compared it to a traditional FODMAP study. Small numbers of patients, as you can see here, only 16 in each group. COVID got in the way of this study. But basically, what you can see is that the results are fairly similar between the 2. This is for the abdominal pain or abdominal discomfort associated with a 30% reduction in abdominal discomfort. This is for abdominal pain. And you see the p-value that was not significantly different, suggesting, but a larger study needs to be done, that FODMAP simple may be the way to go as the initial step in patients. And then if they don't respond, maybe going to a full FODMAP diet. That's what this suggests, but clearly a larger trial might need to be done. So this was just recently published as well. And this looked at the low FODMAP versus a low carbohydrate diet, which is interesting. And they also looked, the third group was the optimal medical therapy. So it's why you see three different groups. And basically, the low FODMAP diet, so 50% of the low FODMAP diet was from carbohydrates, 33% from fat, 17% from protein. And the low carb diet, it was 10% from carbohydrates, 67% from fat, 23% from protein. And again, the amount of FODMAPs that were intake was 19 grams in the low carb and 3 grams in the low FODMAP diet. And basically what you see is that they're fairly similar, the low FODMAP and low carbohydrate, but the low FODMAP is winning out here, and the standard medical therapy didn't seem to do as well. This study, and you'll see this presented a lot, this study kind of suggests that the low FODMAP diet should be the initial treatment option for patients. Because to date, we made that recommendation in the guidelines for AGA, but we didn't have any data to say that diet's first. This study really suggests that diet should be first before we do pharmacological therapy in patients. Now, this is two different studies, because again, they're presented together because they're relatively similar, looking at the role of the Mediterranean diet. So this one is just an observational study. So basically what they did was they looked at patients with IBS. They had 108 patients with IBS versus 108 controls, and they looked at their dietary intake, looking specifically at the role of Mediterranean diet foods that people were consuming to see what correlated with symptoms compared to those that were healthy controls. And basically what they found was that not all Mediterranean foods in that diet are healthy or good for IBS patients. They showed that some of them were quite good, right? But some of them were not so good. So it showed that it really needs to be personalized, and there may be specific foods that can increase symptoms in the Mediterranean diet where others can't. So just by giving a general Mediterranean diet may not be appropriate for every patient. And this is a pilot study, on the right-hand side, looking at the Mediterranean diet versus a usual diet. This is a 6-week trial in patients with IBS, relatively small number of patients. They called it a pilot study. And this is the control group. They were given the Mediterranean diet, but they didn't have IBS. And this is the IBS group. And overall, you can see there was an improvement in symptoms. Interestingly, we don't see many patients getting worse. Maybe that they were unblinded, because it's very hard to blind this, because one would have expected from this study that some patients would get worse, but they didn't. And this leads them to conclude that a larger study needs to be done, but it does suggest the Mediterranean diet may be effective. So moving on is one other area I wanted to talk to you about that's a relatively new concept, and that's the role of food IgG testing. And this just shows you recommendations from all of the allergy societies say that they recommend against the use of IgG testing. Australian, American, European all suggest you shouldn't do it with it. And they conclude there's no credible evidence that measuring IgG antibodies is useful in diagnosing food allergies or intolerance, nor that IgG antibodies cause symptoms, food allergy cause symptoms. We're talking about intolerance, not allergies, of course, with the IgG antibody. What I want to present today is some results of a study that we conducted looking at the role of IgG food testing. So this was not a new concept. This was done several years ago by Peter Warwell's group, where they looked at a large number of IBS patients and 60 plus people in each group, looking at IgG testing and then eliminating the food based on the IgG testing. And the sham group eliminated the same number of foods with it. And as you can see, no difference, but when they looked at people that followed the true diet, because again, most people don't follow, don't do what you tell them to do, but when those that were compliant, there was a statistically significant difference. The issues with this were a couple. One is shown here, is that there was a marked difference in the percentage of foods that were eliminated. So for example, 84% removed milk from the treatment group versus 1% in the sham group. So you don't really know if it's just that dairy is the major culprit, and that maybe you don't need IgG testing for it. The same with yeast and eggs, et cetera. You can see for yeast, 86% versus 0%. So there's really a mismatch. This never really took off, but it was very interesting and intriguing data. So we worked with a company actually out here in Newport Beach that developed an IgG antibody testing. The difference here is based on a large number of healthy controls in IBS patients. So IBS is shown here. This is group of patients. You can see this. You can see the cutoff based on a 95% confidence interval that was developed from the healthy control group. So if you are abnormal, you were really in that 95% confidence interval, and then looked at a large number of foods that were consumed by IBS patients, and then whittled it down to 50. That was still too much, so we whittled it down to ones that were the most likely to cause symptoms. And in this case, it was an 18-food panel that we came up with. And this shows you the results that is being reviewed right now at gastroenterology. And you can see this is an 8-week randomized trial. This was done in conjunction with the FDA, so it was we consider a fairly rigorous trial where we had a sham group that was really based on removing not only the number of foods, but foods from the same class that was removed from the elimination diet food. So we really tried to make sure that they were similar, that it wasn't just like milk, for example, that was the differential between the two. And basically what we found that there was for the decrease in pain in the experimental versus, they did not reach statistical significance, but came relatively close to it. And for the continuous variable, but if you looked at the primary endpoint, which was the 30% reduction in pain based on the FDA responder, you had 59% meeting that versus 42% that met statistical endpoint. And it seemed to be more effective in the IBS-C than the IBS-M. So interesting, maybe not ready for prime time. It is clinically available, but we need more studies to show how effective it really is. But it emphasizes the fact that food may be the major trigger, and it also goes back to what I said initially. Food is complicated. It's not going to be one mechanism for all patients if it's food related. So foods are associated with the development of IBS. I think that's clear. Unprocessed foods, particularly highly ultra-unprocessed food, particularly are involved in the generation of symptoms. Low FODMAP diet is still the most efficacious. Based on that study that compared it to drug pharmacological treatment, it seems to be the first-line treatment agent for IBS, and I'm sure you'll see that in the next set of guidelines that come out. And food IgG testing is interesting, but requires further testing. With that, I'd like to thank you again for the invitation and appreciate your time for listening. Thank you.